Hi all, welcome to the Mod Lab. I'm Desmond, a second year PhD student in CC, studying geological sciences. And I, along with another graduate student and two undergrads, work for our lab's PI, Elizabeth Trembath-Reicher. So we are a newer lab in CC, but we do a lot of really cool research. For the most part, we study microbes that are living below the Earth's surface or deep in the ocean. Sometimes we even look at microbes that are deep below the ocean floor, the best of both worlds, right? So that's why we are the Mod Lab, the microbiology of the deep. So let's start with a little tour of the lab room. We do a lot of different microbiological techniques while we're working in this lab. But for the most part, we need to do all of those in such a way that we, one, protect ourselves from reagents and chemicals that we're working with that we don't want to, mess us up. But we also need to protect the cells and the samples that we're working with. So in the end, we need to work in chemical and microbiological hoods that allow us to work in an environment that protects the samples from cells and protects us from the different chemicals that we're working with. These hoods are where we're going to perform most of our work with cells, right? So if we're going to be staining cells with a fluorophore or labeling them with isotopes so that we can figure out what these cells are doing and how many cells there are and even who they are, we need to do most of that preparation in those chemical hoods. But that's not the only thing we have in the lab. We have a ton of different machines that allow us to do tons of different analyses. For example, this centrifuge allows us to separate sediment from cells. A hybridization oven might allow us to hybridize DNA and RNA to different labels so that we can visualize them in gels. This sonicator produces hypersonic sounds so that we can quickly break apart aggregates so that we can better analyze them in the future. But the last thing that I want to show you in this front room, there's another room, is our microscope. So our microscope is where we're going to do most of our direct analysis. So once we've labeled a cell with a fluorophore or an isotope or some other label to show us where they are, we can look at them under much higher magnification using this microscope. So for example, I actually have a sample of sediments that have been, cells that have been separated from sediments and stained with a fluorophore called DAPI. DAPI is a DNA stain that fluoresces blue, that's these blue dots that you can see here. And so what I'm trying to do with these samples is figure out how many cells there are in specific regions and specific soil samples from an arid area around Tempe. Now, our front room is great, but there are some analyses and some samples that we just can't work with in the front room. And that's because we have to be really, really cautious about contamination, especially with our samples that are taken from deep below the Earth's surface. If I were to take a sample of sediment from around Tempe, right, just a soil sample around here, I'd probably find 10 to the 9th cells per cubic centimeter. But if I were to take a soil sample or a rock sample from deep below the Earth's surface or deep below the ocean floor, I'm going to find probably around 1,000 or maybe even less, 100 cells per cubic centimeter. That's almost 7 orders of magnitude different from what we'd find in a normal soil sample. So in order to prevent ourselves from finding cells that weren't there initially, we have to work in a super clean condition. And that's why we are going to convert this back room into a clean room. It hasn't happened yet because we're a newer lab, but once it does, we'll be able to work with soil samples and sediment samples that have very low cell counts. And without worrying about contamination, without worrying about cells coming from us or from the kind of messy environment around us where microbes are always floating around. We're all really excited for this clean room to get set up, and it's going to be set up soon. But I hope you all enjoyed your tour of the Mod Lab. Thanks a bunch.